This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode 109, recorded on July 30th, 2015. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Or maybe I should say the podcast that studies unseen things using words that cannot be pronounced. Maybe we need to make a new intro for this podcast. Joining me today from Small Things Considered, Elio Schaechter.
Well, hi there.
You remember that quote was from your neighbor, right?
That's right. My neighbor talked about us using incomprehensible words.
Microbiology, the study of things that cannot be seen using words that cannot be pronounced. Someone picked that up and tweeted it yesterday, and everybody loved it.
Oh, really?
Yeah.
That's because the podcast from 2 weeks ago went live this week.
That's right.
Yeah.
And people, there's always a delay in listening, right?
Yeah.
Also joining us today from South Carolina, Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Is it really hot there?
Yeah, it's warm, but we're better than most of the country because we have the sea breeze. They're saying that the Atlantic Ocean is cooler, and the good news, though I don't dare not say it out loud, is they're predicting a quieter hurricane season this year. So that's great news for me.
Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello.
Fresh off the links.
No, I cycle to work. That explains my togs.
How long do you have to cycle? A mile?
Yeah, about a mile.
That's not bad, right?
Yeah, but can I say it's beautiful here in Michigan where there is not a Legionnaire's disease outbreak to my east, unlike you, Vincent?
Yeah, there is one here.
Yeah,
There's one in New
There
York.
is? No kidding.
South Bronx.
Yep.
Wow.
I gave your name to some reporters yesterday. I gave you and Ralph Isberg and Howard Schumann. Are those
Great.
Good names, Michele?
They sure are.
Very good. Yeah, we have a little Legionnaire's outbreak. Michigan does not. But Michigan has other things, I'm sure, which I don't know right now. But
Yeah.
it's
Remember,
really hot.
it was the original. It was Pontiac fever.
It's
True.
Philadelphia, though.
Pontiac fever predated
Yeah, I know, but the
Legionnaires
first
out of
time
Philadelphia.
the first time we picked it up was out of Philly, right?
Yeah, and it's Pontiac fever is a different syndrome altogether, but
Yeah.
That's true.
It's a great name, though. We are in summer here. Hey, look, it's the end of July. Can you imagine? Summer's gone, basically. That's it. So,
bite your tongue.
Yeah. But I'm enjoying it. I'm having a good time. And you know, Michele, I wanted to tell you something. Our book is publishing in 3 weeks.
Wow, good for you. That's great.
Isn't that cool?
Congratulations.
I'm
That's
very
wonderful.
excited. 2 years to review the.
It's a great book, by the way.
Thank you.
Does it weigh more or less?
I don't know. I have to get a copy before I tell you. But I tell you, it weighs less than Mobile DNA 3, which I just got in the mail, which is an ASM Press book. It must weigh 10 pounds. The thing is, I'm looking at it here. It's 4 inches thick.
Wow.
But it is so cool. If any of you out there who are interested in mobile DNA, it is such a cool book. And I didn't realize there were so many different kinds of mobile DNA in all sorts of organisms. My favorite is Sleeping Beauty. You
All
guys,
right, you're
heard
going to
of it.
have to explain that one.
have you heard of Sleeping Beauty?
No.
Nope.
So it was a transposable element discovered in a fish genome, but it was mutated, so it wasn't moving. So they corrected all the mutations, and lo and behold, it came to life and can pop around, and that's why they call it Sleeping Beauty.
Wow.
Isn't that
There
great?
was no prince involved then.
No prints, just good old site-directed mutagenesis, right? That's cool. Anyway, mobile DNA is just awesome. This episode is sponsored by ASM Gap, and I'd like to tell you about a workshop called Turning Your Science into a Company. Have you ever had some ideas during your scientific work that you thought could be commercializable and might lead to the formation of a company, but you couldn't really figure out how to get that started? Well, this workshop is for you. They will have at this workshop successful entrepreneurs who'll tell you how to turn your science into a company. They'll tell you about their stories. They'll give you practical tips, advice, resources, all directed at showing you how to establish a startup. Turning your science into a company will be taking place from October 1st to 3rd. The application deadline is coming up soon, August 20th. And if you're interested in doing this, go to asmgap.org/TSC. And if you can't get that URL from my talking, which is entirely possible, you can go to the show notes for this episode, microbeworld.org/TWiM, and there'll be a link to asmgap.org/TSC right there. All right, today we have a snippet and a paper for you for your summer listening pleasure. Someone tweeted the other day they listened to our podcast at the shore. What a great way to enjoy the weather and be smart at the same time, right? Michael, I will hand it to you for a snippet.
All right. So the paper that I selected for our snippet is got this wonderful title. It's entitled The Precision Guided Antimicrobial Peptide as a Targeted Modulator of Human Microbial Ecology. They had me at the title, and it's authored by a group out of 4 different facilities. The first author, Gao, is at the School of Dentistry at UCLA. The second, Jeff McLean, is in the Department of Periodontics at the University of Washington, Seattle. The third author, Young-Gik Yang, is at the Craig Ventnor Institute. And the fourth author, Randall Eckert, is at the C3 Yan Inc. company in Marina del Rey. And as well as the first 4 authors, there's Chris Kaplan, Pierre Keim, Omid Sheikh, Brian Varnum, Renata Lux, Wei-Yang Shi, and Wei-Song He. And they're at one of those 4 respective institutions. The reason I went through describing where these authors are is because we know that studying the role that an individual species can contribute to the behavior of microbial communities is a challenging problem. We've all seen the great microbiome data coming out, and we've begun to look at all this remarkable sequencing, but consider what we do when we establish authorship for a manuscript. We understand the contribution that each of the individuals makes in authoring the paper. But we can't do that when we study bacteria in mixed or complicated communities. We simply can't ask the individual microbe or the individual species what they are contributing and how they're impacting the effect that we are measuring. So that's effectively the holy grail, if you will, of the microbiome. And now that we got sequencing pretty much figured out and, turning out all these beautiful maps, we now have to figure out what the contribution of individual players are to the community. So today's paper is how the authors are using an antimicrobial peptide, and we've introduced you all to antimicrobial peptides before, and principally what they do is they kill bacteria, but this one is specific because they've learned how to target the peptide to one type of microbe, and in today's paper, that target is strep mutans, and it will eliminate that one type of microbe from the community and thereby enable them to manipulate the microbial community and in turn understand or be able to assess what the elimination of that one particular species has done to the ecology of the situation they are looking at. In other words, using a smart bomb, if you will, to ask what would happen if the classic approach that many of us learned when we were first discovering microbial physiology, where we use knockout mutations to figure out a metabolic pathway, we can begin to, if you will, riff on that principle, but using a different tool, namely to look at the community. So the system that this group...
You should point out that those things don't grow on trees exactly.
Yes.
There are not too many examples of smart bombs in the antimicrobial world, are there?
No, and I just came back from the infectious disease grand rounds where they were talking about bacterial vaginosis, and I'll try to introduce a bit of that discussion at the end because I asked he's a specialist in infectious disease and also a gynecologist who's been treating women for bacterial vaginosis, and I asked him, having just prepared for this, about using a smart bomb to treat yeast infections in women without, resorting to some of the things that are going on. So the first thing that we need to know about is what system that this group elected to study. You can imagine that two of the groups are in dental schools, so you can guess that they're going to look at the oral microbiome. Now, this is an extremely complex and dynamic system harboring over 700 species of bacteria, representing 13 different phyla that are capable of growing at various pHs, electromotive forces, and various levels of oxygenation. So you can well imagine, just like the metaphor I used about the authors, each one of these species or phyla has its role in the paper. And we can guess what the Craig Ventnor group did to contribute to this paper because we're going to talk a little bit about sequencing as well. So for the most part, the environment of the oral cavity is considered to be anoxic and reduced, with many of the microbes being very challenging to grow as pure cultures. And one of the
Would you explain why something which is exposed to the air, like the mouth, turns out to be anoxic?
Principally because many of the microbes in the oral cavity don't have the luxury of using molecular oxygen as their terminal electron acceptor, and so they're constantly looking for places to dump their waste electrons. So any of the facultative aerobes will immediately consume the oxygen that we're breathing in through our respiratory and our oral cavity, and so it gets consumed very quickly. And so the available oxygen that's present at that mucosal surface where many of those microbes are, because we're not delivering blood to them so they can get oxygen, they're effectively being starved for oxygen. And many of the microbes are, of course, growing in biofilms that are very tall, if you will, from a microbial perspective. And so consequently, even if oxygen were able to seep in, it's consumed by the folks on the top of the pile, and the ones on the bottom are effectively starved.
And when you floss, you knock off those biofilms.
And in fact, I had a discussion with a periodontist last week, and the periodontist says, my role in life is to disrupt biofilms. Any of you who have periodontal disease know that you, rather than seeing the dentist twice each year, you see them typically 4, sometimes 8 times a year if you have bad periodontal disease. And what their mission is to disrupt the biofilm to effectively prevent those bad actors from interacting with our immune system, which, the bad thing in periodontal disease is bone loss, where your teeth ultimately fall out. And so the bone recession and the interaction of the microbes in our immune system is the bad thing. So what this group looked at is the most common infection of humans, namely dental caries, which is initiated by Streptococcus mutans. There are an estimated 5 billion of Earth's 7 billion inhabitants suffer from an infection of strep mutans, which ultimately results in tooth decay. And this problem accounts for between 5 and 10% of the total healthcare costs of industrialized countries. So what these workers are doing for us is they're actually attacking a very big problem, whereas if you ask the average person, do you think a cavity is a big health effect? When you really consider how much it's costing each and every one of us, it's really a pretty big deal. And so what Gao and his colleagues have done is they've used this new class of antimicrobials, specifically these targeted antimicrobial peptides, that can achieve target killing of these selected pathogens. And the pathogen they targeted was strep mutans. And they had already worked out this peptide, and they named it C16G2. So rather than saying that over and over, I'm just going to call it the peptide or the guided missile, if you will. And the attributes of the guided missile is, if you recall our discussion on bacterial toxins having an A and B domain, where one of the domains effectively targets the toxin to a specific human cell type, and then the other domain of the toxin does the killing. Well, it's the same principle here. The targeting moiety finds the pathogen, and the antimicrobial peptide is pretty much nonspecific, and it does its killing by collapsing the membrane potential. And remember, when you collapse membrane potential, that generates free radicals inside the cell that accumulate, poke holes in the membrane, fracture the DNA, trash the RNA, and the cell ultimately dies very quickly. And the species-specific binding peptide is the magic. And that's what Elio was alluding to, is they don't grow on trees, but we know how to target peptides to bind antigens, because after all, that's what immunology is all about, the antigen interactions, or the antibody-antigen interaction. And that's the brilliant aspect of this smart bomb, is the ability to target one particular type of strep mutans. And they had previously established that this peptide would kill strep mutans grown in a liquid or when it was growing as a biofilm. And strep mutans is pretty easy to grow in the lab. You typically grow it in a candle jar, so it's pretty easy. And they also established that this peptide will selectively kill strep mutans in a mixed biofilms of another Streptococcus species, sanguinis, and Gordoni. And so those are two streptococci, so they targeted the specificity. Now, this paper builds on that specificity. So the first experiment that they did is they included more oral streptococci species that were closely related to strep mutans, along with non-streptococcal oral species and non-oral representatives. And their first table goes in to how the IC50, or the inhibitory concentration of the peptide, and for strep mutans, the inhibitory concentration is in the ideal range for an antimicrobial, in the micromolar range. And so for strep mutans, that IC50 is 5.9. And so what they did in their first experiment is they established that this peptide had a high antimicrobial specificity against strep mutans. And then they characterized its activity against a laundry list of these bacteria, everything from strep gordonii and strep mitis and strep pyogens and all of these others, to things like E. coli and Pseudomonas aeruginosa and other gram-positives. And what they learned...
You don't want to say other gram-positives. You want to say gram-positives.
All right. I'm sorry. So the way they did that is they plotted an Ic50 plot, and that's generally a semi-log plot with the concentration of the inhibitor or the antimicrobial on the x-axis and the percent of inhibition. And since you measure growth or no growth, it's pretty easy to do. And you get this beautiful S-curve, and the middle of the S is typically at the 50% mark by definition. And what they learned is that strep mutans was 5.9 micromolar, while for the other streptococcal species, the concentration of peptide it took to knock it down 50% was between 5 and 60-fold greater, between about 30 micromolar to almost 400 micromolar in concentration. And it's all about concentration when you think about administering antimicrobials to microbes and other things in order to try to understand what's actually going on. The one outlier that they thought to mention was Salmonella typhimurium, and it was peculiar in that it was inhibited at 23 micromolar, and I couldn't quite make out why a gram-negative like Salmonella typhimurium would be inhibited at 23 micromolar, while E. coli was inhibited at a concentration of greater than 785 micromolar. So it was very perplexing to me. Why? Because I think Salmonella and E. coli sort of look alike on the outside. So there must be something magical about that recognition moiety that's cross-reacting with Salmonella. So they next then addressed how the peptide would perform in a targeted killing of strep mutans within a saliva-derived in vitro oral microbial community spiked with strep mutans. And here's again where these authors were really clever. Rather than using the strep mutans that's running around in our mouths, they used a mutinous strep mutans that had an antibiotic marker, spectinomycin, which is an antibiotic from ancient times. It's an aminocyclitol that closely resembles aminoglycosides, and it binds to the 30S to inhibit protein synthesis. But because they can tag it, they can know for certain that strep mutans in their system is being killed. And so in this in vitro saliva model that they created, they used a medium that had been developed by other workers called SHI medium, which is really nothing magical. It's a combination of proteose peptone, tryptase, yeast extract, 5% sheep blood, and it's able to grow the majority of the microbes in the oral cavity so that what they are modeling is very similar to what's going on in our mouths. So they're able to grow these creatures. So now they have an in vitro system in which they can grow a community like we have in our mouth. They add a version of strep mutans that they can tell if they're killing because of the drug marker. And then they add the missile and ask the question, did it eliminate strep mutans from the community? And the way they did the experiment is, again, very clever. They simply incubated this culture medium for 30 minutes with the peptide. And when they measured it via viability, because they can literally do a viable count, and they asked the question, how many strep mutans grew in the control in the experimental? They learned that in the control, they had 100 million strep mutans, and in the experimental or the peptide side of things, they had 200. That's pretty remarkable. When they asked the question about the other bacteria that are in this mixed microbial community, they had 1 billion in the control per mil, and in the experimental, they had 100 million. So they said, wow, this is really working. And then they did a molecular trick. They used denaturing gradient gel electrophoresis, or DGGE, and they had as their control a population that they added the carrier of the magic peptide. And then on the other control, they had the strep mutans that they spiked the culture with, and then they had the treated. And through DGGE, they asked the question, were they depleting strep mutans? And those of you familiar with DGGE know it gives you a gel picture and a band lights up. And since they have the control, they can actually look at the complexity of the community by looking at these bands. But one of you is going to argue with me, well, how is PCR going to be accomplished on something that is killing it? Isn't the DNA going to still be there? And this is, again, where these authors are really clever. They treated the missile-treated cultures with ethidium monoazide bromide, and that specifically prevents the amplification of DNA from dead bacterial cells and only allows amplification of DNA for their PCR-based DGGE from the viable fraction. So they could
by the way, it's really a clever trick, and it probably is going to be used increasingly. I'd never heard of it. Is this something novel that they did?
No, this has been around.
It.
This has been around for about, I began to see it at posters at ASM probably 6-7 years ago.
I see.
It's really remarkable. But this is the first time that it has real utility because of the smart bomb and the fact that they're able to measure the viable count because of that spectinomycin lesion that they use to spike the strep mutans in the culture. So they asked, and then they could detect an overall shift in the microbial profiles in this saliva-derived oral community recovered after the peptide treatment. In other words, strep mutans is eliminated, and so their next experiment is where the Ventnor Institute comes involved. They go to the pyrosequencing. Now recall that...
You want to go into details on that? Or
yes.
Can we...
Yes.
Well, maybe not.
Well, no. Well, what I was going to do is just give you the take home from what they learned in the teased apart community. The normal community had 21 bacterial genera that could be detected from regrowth of carrier treated samples, and in the experimental where they had the peptide treated, where they depleted strep mutans. They only had 16 bacterial genera that were detected from the regrowth of the peptide-treated samples, with Streptococcus, Granulus satella, and Prevotella being the most dominant ones. And so this is showing us that depleting one microbe can actually alter the population. And that's all I'm going to say about that, because it really illustrates the utility of this technique of using the smart bomb to deplete one actor and seeing actually what it actually does to the global community.
Michael, can you design a peptide for any specific species?
That's what it's implying, and that's the subject of how I found this paper. As I was scanning trends in microbiology, and Jorge Frias Lopez wrote a 2-pager in TWiM, specifically entitled Targeting Specific Bacteria in the Oral Microbiome, and he's advocating designing a peptide for one of the keystone species in the development of periodontal disease. And Dr. Frias Lopez is from the Forsyth Institute, which is in Boston, which is the preeminent laboratory in the world for describing the oral community.
You could use this for other communities of bacteria as well, right?
Yeah, and if in fact, remember, membrane uncoupling will actually kill our cells, it will kill yeast. And so that's where I suggested to the infectious disease gynecologist, I said, could you design a peptide that would selectively target one of the yeasts that is causing chronic bacterial vaginosis without disrupting the other players in the vagina?
And he said he'd love to do that if he could.
Yes, he did. He said that is...
But listen, it's one thing to want to do it, but what's the basis for this? Or what's the rational basis for designing a peptide? I'm not quite sure I know that this is more than empirical studies, empirical playing around with different peptides and finding maybe there are some rules that tell you certain classes may do certain things than others. But in reality, I think it's just a hit and miss thing right now, isn't it?
Well,
is there a rational approach to this?
That's what I had. Since this was only a snippet, I didn't go deeply to find out the answer to your question. And that's
right. But I think I know the answer, and that is that it's not yet possible to design specific peptides against specific bacteria. It's only possible to test them.
And even if we accept that in this particular case, the peptide was specific for mutans, what we see is the ecology playing out, that it actually, you can take that one species out with the peptide, but it brings down 4 other species with them that are codependent because they share metabolic pathways or food chains. So to me, that was the most impressive result.
I think that's the value of this. Right. Besides the possible practical application, the problem with the microbiome is that it's like a telephone book. You don't know where to look for associations, and this is one way to find out that the microbiome consists of sub populations which do certain specific things. You'd expect that. You'd expect all kinds of interactions, but this seems extremely valuable to me.
And as Michele was just taking us through, is the authors then did another experiment that they dubbed their pull-down assay. And again, they're going to use the same methods. Saliva was added to an overnight strep mutans biofilms, and after then 3 hours of incubation, any unattached cells were washed away, and the biofilm was collected either immediately or at time intervals and then incubated in the presence of fresh brain heart infusion. And what they learned is that the strep mutans biofilm was able to recruit and form tight physical associations with specific oral bacterial species, including Fusobacterium periodonticum, Campylobacter, and one Neisseria species, which has a presence that cannot be detected within a community that is recovered from the peptide treatment. And so go ahead.
The take-home message here seems to be that we should really be looking for such things, because not only have the practical effect of being the right approach to therapy rather than a shotgun approach of killing everything in sight, and secondly, they reveal a lot about the structure, the fine structure of a microbiome, which it desperately needs. As I say right now, you want to get out of the telephone book phase and become more aware of what is actually going on in the microbiome. This is just the beginning.
And that's where this snippet ends, because Elio just gave my summary.
Oh, I'm sorry.
No, it's okay.
I didn't mean.
No, but that's okay.
I didn't mean to.
No, that's the whole point. That's the whole point of a snippet. It's to intrigue the audience to go out and learn more. But I think that the key take-home message that Elio and Michele and I have been trying to drive home is we've moved from the telephone book to really understanding the neighborhood and what an individual's contribution is to that neighborhood. Using, if you will, the metaphor I started with, every one of the authors I listed and their 4 respective institutes all made a seminal contribution to this manuscript. They couldn't have done it without the Ventnor group doing the sequencing. They couldn't have done it with the group that made the peptide, and the clinical or translational application that the dentist brought to the table from the University of Washington and UCLA really brought home the ability to really move from this telephone book stage to really understanding how we're going to begin to decipher the wonder that is the microbiome of these various human niches.
As long as we can get peptides for everybody.
And that's, I think, my homework assignment, to stay tuned, as they say. I'll go out and look to see how they design their peptides.
I mean, in principle, it's not hard to get peptides against anything. You just do a screen. But,
right.
You know, whether it's specific or not is another story.
Right.
That's old phage selection, isn't it, Vincent?
I also think it would be, I would bet one day, if this gets more extensively done, that you will see not just 4 other species going down, but hundreds, right? You pick the right one, and there are going to be hundreds dependent on it. That'll be really interesting.
And it may cause more harm than good.
Yeah.
You just don't know.
This is absolutely true, because the other players that would come in are these other streptococci that may have bad actors. And remember, the streptococci have issues in that they can, cause endocarditis. And every time we brush our teeth, we effectively become septic, or we become bacteremic, if you will, for about 4 hours each time we brush our teeth. Just the mere physical abrasion of our gum line drives the microbes from our mouth into our bloodstream.
Yep.
And is it worse if you floss?
Well, according to the periodontist, flossing is the best thing an individual could do, because what flossing does is it's disruptive, and it disrupts that biofilm. That's the whole issue, is the biofilm will mature into plaque and then calculus. And that's what the dentist is always doing to me, is I make a lot of calculus. So they're always scraping the calcium
too much information, Michael. Has anybody done this study to ask how bacteremic we get after flossing?
I haven't seen that. Another homework.
I bet they have.
Yeah, sure.
I'll write it down.
Thank you, Michael. This is lovely. All right, here's a paper on dental flossing and its relationship to transient bacteremia.
You found it already.
Yeah, it's all there, so you can just search for it. All right. Thank you, Michael. We have a paper which I will tell you about. This is published in PLOS Pathogens very recently. It is called the Endosymbiotic Bacterium Wolbockia selectively kills male hosts by targeting the masculinizing gene. I found this a provocative title. I like Wolbockia very much. I don't particularly like killing males, but it's a very interesting study nonetheless. It was done out of the University of Tokyo, several different departments. Fukui, Kawamoto, Shoji, Kyuchi, Sugano, Shimada, Suzuki, and Katsuma are the authors. Now, we have talked about Wolbockia here before. We talked about them on TWiM. Wolbockia pipientis, an obligate intracellular gram-negative bacterium. It's classified in the class alpha proteobacteria. It's a member of the Rickettsiales, which is an order of intracellular bacteria. You can't grow these outside of the host cell. So they're obligate intracellular bacteria. They have to grow inside of a cell. They are estimated to infect about 40% of all, maybe more, or arthropod species. They can also infect nematodes, mites, spiders, and probably many other species that we don't know about. They're very successful, and this is partly because they're transmitted vertically. They start in the stem cells of the ovaries, and then they move to the developing oocytes, and they make it into the offspring. So females transmit the Wolbockia. Males do not. And so this is a good way to ensure that you're going to stay in most of the population. They've been really good at spreading, obviously. And for this reason, we are trying to figure out how to use them. Can we use this to somehow benefit us? And one example is that it turns out that when you put Wolbockia into a mosquito that otherwise doesn't have it, they become less susceptible to viruses, including viruses like chikungunya, yellow fever virus, dengue virus. So lots of people are doing studies where they put Wolbockia into mosquitoes and they try and release them. And because they're transmitted via the females, it's theoretically possible that you could actually get these mosquitoes propagating in the wild. Another interesting example of how we're trying to use them, Filarial Nematodes. These are parasites that cause things like filariasis and river blindness. These nematodes have Wolbockia in them, and they need them. If you take them out, the nematodes die. So you can just treat these filaria with antibiotics, which have no effect on the filaria themselves, but it gets rid of the Wolbockia and the filaria dies. So people are trying to figure out how to use this to treat filarial infection.
Isn't that amazing, by the way? That's really a great story.
It's a great application of basic microbiology.
Yep. But why is it so successful? Well, it's vertically transmitted. That helps a lot. But they also induce a number of phenotypes after infecting a cell that have led to their success. And these include what's called feminization. When they infect a male, sometimes they may develop as a female. They may cause parthenogenesis, which means that females who are infected can reproduce without males. And this is what I understand is the fate of the world one day. Not going to be any men left.
Watch the guy.
We'll miss you.
Thank you. And that's parthenogenesis. Male killing. Males get infected. During larval development, all the male oocytes die, or all the male larvae die. That's what we're going to talk about today. And this, of course, increases the rate of infected females, and that increases the transmission because they are the ones who transmit it. And finally, something called cytoplasmic incompatibility. An infected male cannot reproduce successfully with an uninfected female or with a female that has a different Wolbockia strain. So altogether, these things cause there to be more infected females in a population, and that increases the maternal transmission in the next generation. And you can see that this is a really good, that's why Wolbockia is so prevalent. The cool thing here is that we don't understand how any of these work, so there's a lot of work to do. By the way, Wolbockia also have a very cool phage that's really neat. It's pretty big, and it moves DNA around and has roles in some of these phenotypes. And Seth Bordenstein, who is at Vanderbilt University, talked with us about this on TWiM 332, if you want to learn more about that. All right, so in this paper, they study the mechanism of male killing. And the host they use, the arthropod they use, is a moth. It's called the Ostrinia moth or corn borer. And in this moth, if you infect the moth with Wolbachia, all the progeny are female. All right. So this is the phenotype of male killing is evident in Ostrinia, and that's what they use to study in this paper. Now, a little aside, we have to talk a little bit about sex determination because that's the process that's being interfered with here. In fact, in insects, this has been best studied in silkworm very well. It's Bombyx mori. Some of you may know this is a very well-known experimental system. The way sex is determined in the silkworm, there are two chromosomes, the Z and the W. Females are ZW and males are ZZ. Males
backwards than what we have.
Right. That's right. And I think it's good that males have ZZ because they're always sleeping anyway, right?
Oh.
I'm making fun of men.
Yeah. Well, you had to do it first.
Shut.
But you would love to jump in, right? All right. So on the Z chromosome, there's a maleness gene called, this is wonderful, masculinizer. And we'll abbreviate that mask. Okay. On the Z chromosome, which is males, again, have two Zs, you've got masculinizer gene. If you deplete this gene in male embryos, you get the influence is on another gene called double sex or Dsx. All right, depleting masculinizer makes double sex produce only female type splice variants. All right, so this gene double sex can give rise to female specific mRNAs or male specific depending on how it's spliced. And masculinizer pushes it towards the male. If you take masculinizer away, it pushes it towards the female. All right. So double sex, by the way, is one of these gene that encodes a product that is involved in sex differentiation, which is a complex series of events involving many different proteins. Depending on which ones are turned on, you become male or female. All right. So that's masculinizer, which works on double sex. And then there's a feminizing gene on the W chromosome. Remember, females are ZW. This is called Fem. And interestingly, this gene encodes a small RNA. Now, everyone has heard us talk about microRNAs and RNA interference. Fem encodes what's called a PWI interacting RNA or piRNA. It's another type of RNA interference. It's different from microRNAs, but basically it turns off gene expression. And Fem targets masculine, the masculinizer gene. So if you have a W chromosome, you have this Fem gene, which turns off mask, and you make females. So no matter how many Zs you have, if you have a W, the offspring are male. And of course, if you have no W, then you become a male. So the presence of a W is dominant. And that's because of this feminizing gene turning off mask. So you got all that so far? You okay? It's a little complicated.
But they have a nice schematic
There's a
in
picture
Figure
in there.
6.
And this is an open access article. So if you're listening and confused, you can go to the
or if you want to tell all your neighbors about it, I encourage you to take Figure 6 with you.
All right. Now, why does depletion of masculinizer lead to not having any males? I told you part of the problem is double sex doesn't make the male splice-specific variant, but it also turns out that there's a more complicated issue. If you take away mask, you get male-specific lethality at the embryo stage because mask represses transcription from the Z chromosome globally. And if you don't do that, the expression of all the genes causes male killing. All right. So if you take away mask, there's no repression of Z transcription and the male embryos die. So the authors are figuring this is what Wolbockia must do. They must do something that basically mimics the depletion of mask. You take away mask, you don't make the male-specific splice variant of Dsx, and you don't turn off Z gene expression so the embryos die. All right, that's the background we need. And what they do is they go out into the field somewhere near Tokyo, and they collect about 80 Ostrinia moths. So that's the fun part, I think. And you catch them, you make sure it's Ostrinia. You can tell by the way they look, and you can do PCR to make sure it's an Ostrinia. Out of these 80, they found 3 females that had Wolbockia in them. And they have a lot of cool technology here. So how do you know it's a female? They do PCR, and they look for the Z. They look for the Z-linked genes, right?
You can't tell them apart?
it's probably not so easy, and that you may get an idea, but I can't tell male and female mice apart, so I have to do PCR. I used to separate the males and females, and then I'd go back and they all had babies the next month, So obviously I couldn't do that very well. All right, so they found 3 females with Wolbachia. When they mate these with Wolbachia-free males, only females are born. All right. However, if you look in the eggs before they hatch, or the embryos before they hatch, you can find both male and female larvae. And you know that because, again, you can do PCR and ask, is this a male or a female, depending on the ZW presence, right?
Could I back up? Because I was really impressed by the penetrance of this phenotype.
Yeah.
You said that no males were produced.
Right.
What they did was they mated the 2 types of parentals for 5 generations.
Right.
They generated 302 females and not a single male.
That's right.
So it's really a strong mechanism.
No males. Yeah, no males
Good
at all.
point.
All right, so only females are produced, but the embryos are both male and female, so they're dying before they hatch. And finally, if you treat Wolbachia-infected females with tetracycline and you mate them with Wolbachia-free males, then you get males and females produced. So the Wolbachia is doing what it's supposed to be doing. So the next thing they did is Dsx there. Dsx, or double sex, is the gene that can produce either a male or female splice variant. In uninfected female moths, they can find both male and female type Dsx splice variants, which is what you would expect. But when you put Wolbachia into the female moths, you do not see the male type Dsx splice variant. All right, so this is a clue about how Wolbachia is manipulating sex. It's somehow repressing the Dsx splice variant. And if you remember, I told you earlier, the masculinizer, this gene that's important for producing males, it acts on double sex to produce the male splice variant. If you take away masculinizer, there's no male-specific splice variant of Dsx. So Wolbachia is mimicking this in some way. So that's now the question. How does Wolbachia manipulate Dsx? So first, they have to start from the beginning with these moths because they don't know a lot. They have to say, is there a mask gene in this particular moth? So they have to identify it and sequence it and so forth. And they find a gene with substantial homology with known mask genes. And then they take cells. They use actually silkworm cells initially. And they take this Ostrinia mask gene and they put it into these silkworm cells. And that turns on the production of male-specific Dsx. So again, Dsx gene can give rise to male or female splice variants of this gene. When you put the Ostrinia mask gene in, it turns on the male-specific one. So that's evidence that this protein has masculinizing activity. All right, so now we have a mask gene in Ostrinia. We have a Dsx gene as well. So then the question is, what does Wolbachia do to mask RNA in embryos? So they look at the expression of mask mRNA. In the absence of Wolbachia, this mRNA peaks at 6 hours before egg laying. So remember that the females lay the eggs and then they develop and then eventually they hatch. So 6 hours before the eggs are laid, the mask mRNA peak and then it starts to go down. So at that point, mask is involved in sex determination. So it's important that it be there at that point. If you have Wolbachia in the female moth, by 6 hours, mask is already going down. And then it remains at very low levels. So somehow the Wolbachia is downregulating the mask gene. Therefore, you get no male splice variant of Dsx, which you need to get males produced. Now, remember, the other effect of removing mask is that you get expression of a lot of genes on Z, and that's lethal. So they said, does this happen in Ostrinia as well? So they use RNA sequencing to see if you deplete mask mRNA by Wolbachia infection, which I've just told you happens, what happens to Z gene. And they find that, in fact, Z gene, the entire Z chromosome, all the genes are overproduced in the absence of mask, which is induced by Wolbachia, and the embryos die. So this is the same thing that was observed previously for silkworms. Okay, so the presence of Wolbachia downregulates mask, and that turns on genes on the Z gene, on the Z chromosome that are lethal. And finally, the last experiment, they take mask mRNA. They produce this in vitro. They inject it into Wolbockia infected embryos. Then you get both male and female larvae produced.
What a great experiment that was.
That's
really that's so neat.
That must have been a happy day in the lab.
In the control, they put an mRNA for GFP. Only females are produced. So mask is determining the sex, and Wolbachia is somehow causing a decrease of mask RNA. You get aberrant Z gene transcription, embryonic death, and failure to produce the male-specific Dsx. So the next step, obviously, is to identify what it is in Wolbachia that is decreasing mask mRNAs, and they suggest that it could be an inhibitor of transcription, or it could be something that is degrading the RNA. So I was trying to think of how they would do this, and
with an obligate intracellular pathogen,
right?
Where you don't have a lot of genetic tricks at your disposal.
So I mean, if you could, if you really had good genetics, right? You could do. Tell me if this is wacko. You do a transposon mutagenesis and then you put all the offspring in and see which ones lose the phenotype. But maybe you can't do that with Wolbachia.
But is it the Wolbachia because you do the transposon in the Wolbachia and then you infect and then you see what happens?
Yeah, be tough. But what other approach could you do, right? I mean,
I just don't know how you're going to be able to do the transposon mutagenesis since it's an obligate intracellular parasite.
You can't grow it in, but you could grow it in cells in culture, right? Intracellularly.
Yeah, and then I guess you could fish it out. But how would you get? It's not like you're going to get a clone where you can pull an individual colony. Because what you want to have happen is natural selection to see the desired phenotype that you want to see.
Yeah. No, that's a tough one. I agree. The alternative is to make extracts of Wolbachia, and then if you have a transcription of mask, let's say in vitro transcription system where mask is made, you could add those. If it inhibited, then you could try and purify what is the factor that's doing it.
Or genetically, you could identify those organisms that can do only the male-specific splicing or female-specific splicing by putting either a selectable or a counter-selectable marker downstream.
I think I have it, Vincent. You said it had a phage.
Yes.
So what you do is you change the phage recognition element that targets where the phage inserts into the Wolbachia genome, and the phage becomes the equivalent of your transposon, like the old mud phages.
So you would make it so it could integrate anywhere, basically.
Yeah, integrate anywhere, and that would, because you would add the phage would get into the intracellular host and be able to do its act, and then you would just see what would happen.
Parenthetically, that's an interesting issue. How does the phage get into the host and get into the Wolbachia? I asked Bordenstein that, and it's something that they really want to know, right?
Well, the same instance with phage of mycoplasma and chlamydia and all these other intracellulars that have phage.
All right, so that's the paper, and that is how Wolbachia, at least in an Ostrinium moth, kills all the men.
So I double dare you to make a connection between this paper and this snippet that Michael described.
We're specifically killing men, and we're specifically killing a species of bacteria. I think.
Oh wow, that's
it's
good.
a smart bomb.
Precision.
Now turn that into a title, and we're done.
Precision killing.
It's the smart bomb.
Here it is, Michele. Precision killing of men and bacteria.
Yes.
Very good.
Instead of taking the men out of the population.
Male, but not men.
Male?
Males.
Male? Yeah, okay.
Because these are moths.
Right. Moths can't
nice
be
paper.
men, right?
Wow,
it's a lovely
that was a
paper.
great paper.
And the thing is, that's very cool. I didn't go into it too much. They use all the modern technology. They're doing deep sequencing, PCR, RNA sequencing. It's really neat on a neat organism.
Yeah, which... these relationships would have been really difficult to tease apart, 10, 20 years ago.
Yep.
It's really beautiful work.
Yeah.
Did you learn anything about that, Michele?
Yeah, I have not been following the Wolbachia biology. I know that in general, I knew about the killing the males, but this is very cool.
All right. Let's do a couple of emails. We actually have an email that is related to Michael's snippet. And this is from Jeffrey, who writes, I just went to my semi-annual dental appointment, and I thought of a potentially interesting oral health monitor, the plaque biofilm that they scrape off your teeth during the cleaning. Brush immediately before the cleaning to remove the loose food bits, then send the scraped-off plaque biofilm for sequence analysis. It wouldn't have to be deep sequencing, only to monitor the presence of, say, 20 common plaque microbes. Obviously, detection of dangerous pathogens would trigger a return visit, but I am thinking that the plaque biofilm is probably a fairly stable and person-customized biome, so any serious changes to the biome would trigger further dental investigation. Do you folks have familiarity with dental plaques, or could you get a guest on? What kind of sequencing testing might be effective and affordable for this kind of monitoring? Do you have any ideas, Michael? Is this reasonable?
The plaque biofilm is people have been characterizing it. Whether or not it would become a chairside test really is a function of how cheap sequencing will become, and whether or not you could. It's almost like some of the diagnostics that the clinical labs have developed for upper respiratory viruses, where they swab your nose and they tell you whether you have enterovirus or cold virus or whatever virus. Right. And I think it would be something like that we would see first before we would see, one of these sequencing where we would have a potential of 700. But, things are changing every day.
So he's thinking about doing this in your home, right? Because you could do it on a weekly or monthly basis to see if there's any. I mean, because if you only go twice a year to a dentist, then, having it done there would not be very informative.
And that study is pretty controversial as well. Is there any real clinical benefit of going to the dentist twice a year?
Yeah. As always, I enjoy your show, and my special thanks to Dr. Swanson for allowing a fanboy to visit her back in April, I believe.
Oh, how about that?
You remember?
I do.
All right. Next one's from Mark. Hello, current TWiM cohort. I've enjoyed your recent additions of Talmudic questions into show episodes. I was surprised one of your listeners wrote in saying, and I paraphrase, that using the word Talmudic was introducing unprovable theology into the show and veering away from science.
Oh, boy.
Growing up and studying on the East Coast, I came to understand Talmudic with a lowercase initial t as an adjective. It means being roughly the study and making specific distinctions between things relevant only to expert. Written with a capital T, the word becomes a noun meaning study or interpretation of the Jewish Torah. The word Catholic, lowercase, Catholic, uppercase, is also an adjective or noun depending on the case of the initial C in the word. Spoken word podcasts don't differentiate case and can be ambiguous. In a recent episode, there was discussion about the human immune system, its memory, and loss of memory by B or was it T cells. This anthropomorphizing raises several interesting and, dare I say, Talmudic questions. And he gives a list of some Talmudic questions relating to the immune system, like, how did the immune system evolve to have different memory ability for different pathogens? How does the immune system lose memories? Isn't the ability to recognize and fight non-self pathogens a benefit? Is there a beneficial function by memory loss, i.e. the immune system losing memory about some pathogens? Is immune system memory loss a result of pathogens that have a near homology to the self's own protein? What is the essence of the immune system's memory? Is it the structure and shape of a pathogen's proteins? If spoken or written fiction creates human memories, then is there a corollary in terms of the immunity system? Can we fabricate different shaped proteins to create immune system memories against real or imagined pathogens?
That's the holy grail.
Yeah, we would like to know to do that, wouldn't we? Is the cytokine storm analogous to the immune system using dumb bombs instead of precision laser-guided munitions?
Here's our connection to today's story. I mean, cytokine storm is really a dumb bomb.
Yeah, cytokine responses
a scatter bomb.
Are
a scatter bomb, like a grenade.
And a storm it's just not regulated, right?
No.
Because you scatter and then you turn it off, but in the storm you don't turn it off. Is there an immune system corollary to post-traumatic stress disorder, i.e. does it suffer dysfunction resulting from memories of past activities?
I think that's autoimmune disease, isn't it?
It could be. Yeah. I offer these up as Talmudic small t questions for you, TWiM hosts, and your listeners. If any of these questions have answers, please explain and strike them from the list. The journey is the reward. I think how the immune... Well, many of these we don't know the answer to.
No. They're
That's
Talmudic.
That's the point. If you had the answer, it's not a Talmudic question.
Yeah.
As we use it.
And each one can generate a lot of conversation.
In fact, memory is a very hot topic in immunity right now because we're trying to figure out how it works. And, there are some pathogens that actually manipulate memory. In fact, I found a paper while I was looking for today's paper. I think it was a Borrelia paper about, no, maybe that's something else. Anyway, pathogens do manipulate memory for sure. But, memory is basically long-lived B and T cells. And that's the essence of it. And sometimes some pathogens stimulate them and others do not. Anyway, I don't think we could...
I'm really glad that this came in, and thank you.
You can use some of these, Elio, in your blog.
We should, absolutely.
Yeah, for sure.
Absolutely.
You got 9 there you can go on. I know you're not an immunology blog, but it's okay.
Why not?
Right. The next one's from Jim, who writes about a program from the Canadian Broadcasting Corporation. He thinks we might be interested. It's called Science Under Siege. And the description is, scientists around the world are increasingly restricted in what they can research, publish, and say, constrained by belief and ideology from all sides. What happens to societies which turn their backs on curiosity-driven research? Well, I can tell you that they're all gone.
Yeah.
I'm just kidding. But I think
if
you don't have curiosity-driven research, you're in big trouble.
The analogy is akin to the person who cut the last tree down on Easter Island.
Yeah, so that could be interesting. Check that out. And let me read one more. This is from a person whose ilk we've been meaning to have on TWiM for some time. This is from Edwards, who writes, "Hello, TWiMers from State College, Pennsylvania. As most residents would agree, summer is what earns this place the name Happy Valley. With plenty of warm weather, lots of festivals, and plenty of options for outdoor recreation. This summer, we also have more rain than we know what to do with. Sorry, Elio." Hey, Michele, they probably call where you are Happy Valley too, right?
Well, we're not really a valley.
Okay. But you have lots of festivals, warm weather, and options for outdoor recreation.
Yeah, it's a happy college town for sure.
I was driving to my sister and brother-in-law's cabin outside of Syracuse this 4th of July, and my inner nerd was more than happy to make this drive a total TWiM binge.
Right.
Let's just say I listened to many, many episodes. I was on Route 13 making the climb out of Ithaca, New York, with a beautiful view of Cayuga Lake on the left, listening to episode 105 when Michael made this ASM Division P chair smile with his food microbiology discussion about E. coli and salmonella contamination on fresh herbs. That giddiness was short-lived, though, as he wrapped up the story saying he learned growing up the old adage that you should eat local and not global, implying that the globally sourced herbs from this study were the reason behind the levels of contamination. Luckily, I was past Cayuga Lake by this time, as I may have driven off the cliff that leads to it otherwise. There are several reasons to eat local, including to support your local economy and its hardworking farmers, and to eat produce that is as fresh and tasty as possible. Unfortunately, the myth that local means safer perpetuates in the minds of many people. My colleague, Dr. Catherine Cutter, and her talented graduate student, Joshua Scheinberg, for example, highlight several outbreaks from small vendors selling strawberries, bagged peas, and cashew cheese, as well as known hazardous foods that are mistaken to be safe because they are local, such as raw milk. Kathy and Josh have also shown that chickens purchased from Pennsylvania farmer markets have a much higher incidence of salmonella and campylobacter than store-bought chickens. To food microbiologists, this is not terribly surprising in retrospect, as the big factory farms have armies of microbiologists developing strong pathogen control programs, and these companies strive to ensure that their products are refrigerated continuously from the plant to wherever they end up across the U.S. Unfortunately, many in the press spun this paper as implying chicken from farmers markets isn't safe, when the more accurate conclusion stated in the paper was that these vendors need proper training to minimize the risks to the purchaser. The smaller clientele base, limited distribution area, and CDC estimates that only 1 in 30 foodborne illnesses caused by high-profile organisms, such as E. coli, campylobacter, and salmonella, get reported, means that the larger food producers and distributors give epidemiologists more data points to confidently identify an outbreak. This is a better explanation for why more foodborne outbreaks covered by the press are linked to larger companies. You also correctly stated that washing produce reduces pathogen load, but it doesn't eliminate it, and mentioned talks on this topic at the 2014 ASM. This advice should also have mentioned the presentation at the same meeting, but by Dr. Maria Brandl. Her group at the USDA and others showed that human pathogens are not terribly happy on the dry and nutrient-limited surfaces of a vegetable leaf. However, damaged by harvesting equipment, plant pathogens, or senescence can cause plant cells to release their banquet of nutrients, enhancing the growth of non-native bugs such as E. coli and Salmonella. Therefore, another recommendation is to be wary of older produce or that with obvious damage. Thank you for what you folks do. Keep up the outstanding work. And Michele, let me make a prediction for November 21st, 2015, Penn State 35, Michigan 27.
You're on.
So Edward Dudley is an associate professor of food microbiology. Anybody know him?
No. I do not.
Now we do.
Now we do, but
some good points.
That was a really thoughtful discussion of a complicated issue, isn't it?
Yeah, so I think we shouldn't dismiss locally grown produce. I think he's got some good points.
Well, I think we all have to just be mindful of how we should be washing and preparing our food, no matter where it comes from.
Right.
And look at it. I think that's the other take-home message. You look for the damaged leaf and take it off and hope that it hasn't contaminated the rest of the food.
Right. Well, Michael, you should be glad that you did not cause him to run into...
and to run off the lake. No,
Cayuga.
but I think the whole intent of that paper was what they learned is that the herbs that were indeed transported from that area of the Middle East did indeed have a higher incidence of contamination. And it was the same situation that I think he's bringing up in his discussion about the local small individual, because many of these herbs were cottage industry. They were literally families that were growing them together and they were collecting them. And he's absolutely right. And, that's one of the things I always have been so proud of the USDA, and that our food is indeed so saved. And it's principally because of folks like Dr. Dudley and all the folks working in the food industry and the USDA that keeps our produce and meats in a safe condition.
All right, that has been TWiM 109. You can find this at microbeworld.org/TWiM. You can also find it on iTunes, and if you have an Android or iOS device, there are plenty of podcatchers that you can use to subscribe for free. And if you do have questions or comments, please send them. Send them to twim@twiv.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
What a pleasure.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, Vincent and everyone.
And I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the American Society for Microbiology for their support of TWiM, and Chris Condayan and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can hear his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.